Okay, so we're going to be looking at Chapter 22.6, Natural Polymers. So our learning objectives are going to be understanding proteins, carbohydrates, and nucleic acids. Okay, so for proteins, there are fibrous proteins, which provide structural integrity and strength for many types of tissue and are the main components of muscle, hair, and cartilage. And that's seen here, and you can see how the structure is kind of tightly bound, which shows the strength. And then there are globular proteins, and their roughly spherical shape makes them the worker molecules of the body. They transport and store oxygen and nutrients, act as analysts, fight invasion by foreign objects, participate in many regulatory systems in the body, and transport electrons in the process of metabolizing nutrients. And those are seen here on the right, and those you can tell that it's a more spherical shape. Okay, so amino acids. Amino acids are considered the building blocks of all proteins, and in the structure, R can represent H, CH3, or a more complex substance, and the amino group NH2 is attached to the carbon, which is seen here, and it's attached to the carbon, and then which is next to CO2H, which is over here, and then here's the R side chain down below. Alright, so polar versus nonpolar side chains. The distinction between polar and nonpolar classes is determined by the R groups, also known as the side chains. Nonpolar side chains contain mostly carbon and hydrogen atoms, and then polar side chains contain mostly nitrogen and oxygen atoms. And then when you think of polar side chains, they're hydrophilic, which means that they're water-loving, and then nonpolar side chains are hydrophobic, which is water-fearing. So when you hear phobic, it's kind of like phobia, which is a fear of something, so that's how you can know that it's a water-fearing substance. Okay, so here are some polar R groups where there's the nitrogen and oxygen side chains around the amino acids. And then here are nonpolar groups where there's the carbon and the hydrogen in the side chains. Okay, so aqueous solutions of amino acids. Since aqueous solutions can have both polar and nonpolar groups, they can act as both a weak base with a conjugate acid or a weak acid and a conjugate base, and that is pretty much a buffer solution. So optical activity. Optical activity is due to two factors. First, the asymmetry of the polypeptide chain, and second, the asymmetry of the alpha carbon atoms in the amino acid residues. So kind of to simplify that, the amino acids that contain a carbon atom, tetrahedral carbon atom with 4 different groups attached, are considered optically active. So polypeptides. The structure above is called a peptide linkage, and addition condensation reactions lengthen the chain and produce a polypeptide. Eventually, this would yield a protein. A peptide bond is a chemical bond formed between two molecules when the carboxyl group of one molecule reacts with the amino group of the other molecule. A tripeptide sequence contains 3 different amino acids. So for this one, there's alanine, glycine, and serine. And then the shorthand notation of this, like the 3 amino acids combined, would be these 3 word codes down here. And then for a tripeptide, there would always be 6 different sequence options. So here's the tripeptide reaction between alanine, glycine, and serine, and the reactions occur here at the nitrogens between each of the amino acids. So proteins. Any of a class of nitrogenous organic compounds that consist of large molecules composed of one or more long chains of amino acids and are an essential part of all living organisms, especially as structural components of body tissues such as muscle, hair, collagen, etc., and as enzymes and antibodies. So the monomers of proteins, or that make up proteins, are the amino acids. The primary protein structure, this refers to the order or sequence of amino acids in the protein chain. 3 letter codes for the amino acids are used to indicate the primary structure. And so here's that shorthand notation as we saw earlier to represent the lysine, alanine, and leucine here of the amino acids. Secondary protein structure. The secondary structure is determined to a large extent by hydrogen bonding between lone pairs and an oxygen atom in the carbonyl group of an amino acid and a hydrogen atom attached to a nitrogen of another amino acid. So interactions occur in the coils, so like here, in the coils to form a spiral structure called an alpha helix. Alright, so the tertiary protein structure. This is maintained by several different types of interactions, including hydrogen bonding, dipole-dipole interactions, ionic bonding, covalent bonds, and London dispersion forces between nonpolar groups. The amino acid cysteine helps stabilize the tertiary structure of many proteins, and here's that amino acid seen here. Alright, so denaturation. The three-dimensional structure of a protein is crucial to its function. The process of breaking down the structure is called denaturation. If a protein loses its structure, it will not be able to fit inside the correct enzymes, and thus won't be able to function properly.
Next, DNA versus RNA. DNA is a long polymer with deoxyriboses and a phosphate backbone. RNA is also a polymer with a ribose and phosphate backbone instead. DNA has 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. RNA has 4 nitrogenous bases: adenine, guanine, cytosine, and uracil instead of thymine. After that, we have the nucleotides. Now, a very commonly asked question is, what are the 3 parts of a nucleotide? Well, first, we have the nitrogenous base. And purines and pyrimidines are the 2 categories of nitrogenous bases. Second, we have pentose sugars. DNA pentose sugar is 2-deoxyribose. RNA is ribose. Both are 5-carbon sugars. And third is the phosphate group. The phosphorus atom is the central atom of this group. The base and sugar combine to form a unit that reacts with phosphoric acid to create the nucleotide. And this is illustrated in our images. Next, we have double helical structure and base pairings. The key to DNA's functioning is its double helical structure with complementary bases on the 2 strands. The bases form hydrogen bonds to each other. Note that the structures of cytosine and guanine make them perfect partners for hydrogen bonding, and they are always found as pairs on the 2 strands of DNA. Thymine and adenine form similar hydrogen bonding pairs, as shown right here. Look at the colors and look how there's a double strand and how they all bond together. That is DNA. DNA replication. The 2 strands of DNA unwind during cell division, and that new complementary strand is constructed on the unraveled strand. Because the bases on the strands always pair in the same way, cytosine with guanine and thymine with adenine, each unraveled strand serves as a template for attaching the complementary bases, along with the rest of the nucleotide. This process results in 2 double helix DNA structures that are identical to the original one. Each new double strand contains 1 strand from the original DNA double helix and 1 newly synthesized strand, making a base for new DNA. Genes and codons. A gene is a given segment of the DNA that contains the code for a specific protein. They transmit the primary structure of the protein, which is a sequence of amino acids, to the construction or machinery of the cell. Now, there is a specific code for each amino acid in the protein, which ensures that the correct amino acid will be inserted as the protein chain grows, much like an assembly line. And a codon consists of a set of 3 bases, as shown in our image below: guanine, cytosine, and adenine in 1 codon. Finally, we have our protein synthesis. In this, messenger RNA takes the unzipped DNA and delivers it to the transfer RNA. Using an anticodon, a triplet of bases complementary to the codon, the transfer RNA decodes the message from the messenger RNA. It converts the codons from the messenger RNA to amino acids that are added to the protein chain.
In mutations, why is the changing of a single base the least serious form of mutation? Well, addition and deletion mutations generally produce non-functional proteins or none at all. They are in fact frame shift mutations. A frame shift mutation alters the reading frame of the DNA sequence and changes all the amino acids in the protein product after the point of mutation. Substitution mutations only replace one base with another, and because the genetic code is redundant, substitutions will have no effect at all. Substitutions that replace one amino acid with another vary widely in their effect depending on the substitution and its location in the amino acid chain. A substitution that produces a stop codon, or AUG, is the most serious as it will end in an amino acid chain prematurely. Other substitutions can have severe effects if the replacement of amino acid radically changes the shape of the protein or the active site. Changes that have minimal effect on the protein configuration are less likely to dramatically affect the functioning of the protein. Carbohydrates is a large group of organic compounds naturally occurring and includes sugars, starch, and cellulose. They are polymers composed of monomers called monosaccharides, or simple sugars, with the monosaccharides being polyhydroxyketones and aldehydes. This monosaccharide on the bottom left is called glyceraldehyde, and it is not optically active because it cannot be flipped in its mirror image. Disaccharides is any class of sugars whose molecules contain two monosaccharide residues, and glycoside linkage is the elimination of water to create a carbon to oxygen to carbon bond to complete a ring. And glycoside linkage can be used to create a disaccharide sucrose formed from glucose right here. And as you can see, the hydrogen bonds to the oxygen, creating a hydroxide, and the oxygen and the carbon bond to create a single bond, completing the full chain. Starch is a polymer of alpha-D-glucose, and it consists of two parts: amylose, a straight-chain polymer of alpha-glucose, and amylopectin, a highly branched polymer of alpha-glucose. Starches are water-soluble because of the hydroxyl groups involved in the polysaccharide chain. As you can see in these foods, they all break down in water because of the water-soluble. Cellulose is a polymer of beta-D-glucose. Note that the beta-glycoside linkages here in cellulose gives the glucose rings a different relative orientation that is found in the starches. And lastly, glycogen. Glycogen has a similar structure to that of amylopectin, but it has much more branching here.